5 of What is Life in chapter 4 of our textbook. So what we're going to explore right now is really one of the most incredible aspects of the cell. It's the powerhouse, the nuclear power plant, whatever you want to call it. It is where all of the energy is derived, the mitochondria. Mito work, chondria is sort of a organelle that is dedicated. Now, this did not come from the particular cell itself. We talked about endosymbiosis. This was at one time a free-living organism that specialized in forming ATP. And if you see the design, it's a bag within a bag. And with that design, really what you have is a battery effect across the membrane. I'm going to say that many times because it is a tricky concept to grasp, but what we have is a difference in hydrogen concentration from the outside here in this inner membrane space versus the inner membrane and the fluid inside there. And that difference provides a battery effect. There's more charge on one side versus the other. That's a battery. And we're going to talk about how that actually drives the system. But that may or may not be the way you were taught about mitochondria, but that's really what it is. It's a little machine, a nano machine. And so we'll talk more about that. You saw a function of that. There's some videos that I provided with the inner life of the cell is one of them, and it was a series that was performed with Harvard University. A really quite interesting series. It really changes your perspective of what these cells are and what they do. It's a free living. It moves. Every time you've ever seen a drawing of a mitochondria, perhaps it was static, so you would think it just sits there. That's farthest from the truth. And so within the milieu of the cell are these mitochondria that park themselves at places where energy is needed for the normal processes. And if we start to go and find one of these inside this mitochondria, we see all sorts of pieces and parts and various things, and then you start to recognize there's something going on. And what you recognize is these little motors that are spinning inside the cell. And these motors that spin, actually, here's the hydrogens that come across. This is the battery effect across from one side to the other. And the hydrogens are the ones that are transferring, going from an area of higher concentration to an area of lower concentration through a membrane. And as it goes, it provides this momentary sort of attraction due to the charge of the protein, and the positive attracts. It also, if you have a sort of a positive charge as part of the infrastructure, it also pushes. So you have an attraction and a push. If you've ever played with magnets, you can flip it around and it attracts, or you flip it the other way, it repels. And the net result is this starts to spin. Every time a hydrogen goes through, it just adds the next little emphasis for it to start spinning. And so in your mind, if you can think about this thing spinning, and then these subunits up here actually holds the ADP and inorganic phosphate and puts them in such close approximation to one another that the energy it takes to transfer that inorganic phosphate is very, very low comparatively. So you've reduced the amount of energy it takes, and you do this on a cyclic or this spinning motion, and it's sustainable. You could make lots of them. It's like you're just pumping them out, sort of like the Ford assembly line. It's just going as fast as that can spin. You're making ATP. And this is sort of like a little stator, which means that it stays firm, and it forces in these subunits to force that close union between the inorganic phosphate and the ADP to make ATP. So you can see that as it spins around, there's inorganic phosphate and ADP, and as it spins, it it crunches these two so close together. The amount of energy it takes is so low that we form ATP, and the rotation just keeps going and going and going. And now you can kind of appreciate a sort of, here's, you can see the animation. It's really impressive, I think, to know that these little machines, billions and billions of these in your 100 trillion cells, are in there spinning and working and doing all sorts of things. So let me prove that to you. And I will run it here. Hopefully. There we go. And there's the mitochondria. And this is just a video. So you can see the spinning of the ATP synthetases. Isn't that amazing? Every time it rotates, it puts the inorganic phosphate close proximity and makes the ATP. And then that goes throughout the cell wherever it needs. Pretty cool, huh? So let me go ahead and kill that, and I'll come back hopefully to our slide that hasn't yeah, if you want an iPad, feel free. I'm selling one. You can find that on eBay. Isn't that amazing? I just threw an ad in there. Anyhow, so there's the ATP synthetases. They always appear in eukaryotic systems as a pair, and I find that interesting. But again, sharing of common resources, there's nothing is ever by chance. It's there's reasons for all of that, and it's a magnificent drawing that Mr. Astrakhan had done with his company Zyvo. It's just I think this is the next stage. Anyone that wants to pursue in scientific animation, I think you're going to have a heck of a career moving forward with all the new technologies, the augmented realities, virtual realities, and all that. And I can connect you up with Zyvo if you're interested in pursuing a career like that. And I'll see if I can get you to talk with Andrew Astrakhan, and CEO of Zyvo. He started by spray painting shirts, t-shirts, you know, with those little canister things at malls and things, and now he's a multi-millionaire with his company that's doing amazing animations. And it's just it's an amazing thing. If you're interested, you have that talent. I would like to make sure that you get where you need to go, and see if you can move in that direction. So again, it's ATPase is sort of the slang or the short version, but it's ATP synthase technically, as it is an enzyme because it catalyzes a reaction, but it happens to be cyclic, and that's really cool, I think. And again, the rotational is where we win, and that's how the energy is sustainable. And so the number of mitochondria in cells vary. The number of mitochondria per cell varies with cell type. So liver cells, now, you know, you may or may not be aware of what the liver does. It is the detoxifying organ of the body. It's the only organ that both fresh oxygenated blood and deoxygenated blood is supplied to it. So the chemistry that's involved requires certain levels of oxygen tension and those sorts of things. It's an amazing organ. And you get cirrhosis of the liver, it's bad news, from drinking or other types of things that you probably shouldn't be doing. And ultimately, the liver is so important. Skeletal muscle, you can imagine, utilizes a huge amount of ATP, depending on how much exercising you do and that sort of thing. White blood cells in the lung, there's a lot of activities, of course, of breathing in. You get all sorts of contaminants and things. So your immune system is very active in the lung. That's why you react to allergens so quickly, because we have so many immune cells. And of course you're breathing in air, so pollen and all that causes problems for breathing. Just under the skin, the dermal cells, white adipose, depending on how much fat storage. So my number would be about 100 million, but most of my students would be sort of in the range of 100. And then absolutely no energy is being utilized in the red blood cell. It's a chemical reaction with oxygen and deoxygenation, and it moves around by the pumping of the heart. So that goes back up to muscles. It's not a skeletal muscle, it's a smooth muscle, but anyhow, it's really cool to see this. And we have so many more cells that have such a need for the mitochondria. So it's almost like a portable little generator that we have to push around. Speaking of which, if you're interested in losing weight, now ladies, it may not find it as appealing, although the same concept works. If you were to build up the largest muscles that we have, of course we have the gluteus maximus muscle and the various muscles of the leg. If you do a lot of squats and running up and down the bleachers or something like that, you build those muscles up. So my point is, you build up the largest muscle group, and just by the fact that you have to maintain that muscle, it's like these diesel trucks down here, as you were wondering why I put those down there. If they're all idling, they're burning just a little bit of fuel, diesel, just a little bit, to keep the trucks idling. And a lot of truckers do that because you can see their overnight cabs and stuff like that. And you just let it idle. So the more muscle mass that you have, the more of these trucks, because the mitochondria have to support that infrastructure and however much of that mass that you have. So if you build up those muscles, you'll burn more energy and utilize ATP, and hence burn a lot of the fat, and you lose weight. And you're really not exercising once you develop the muscles. Just the maintenance of it helps you increase your burn rate and helps you lose weight. So I just thought I'd throw that out. Now ladies, this is just for the ladies. There is a phenomenon associated with men that they tend to lose weight faster than women, and it's pretty well known. And I had a part of my life after our two kids, I tended to gain a lot of weight, so I needed to lose it. So I did. I lost a fair amount of weight in about 8 months. It's amazing what you can do eating kale and rocks the entire time, but I won't go there. But the idea is that men will lose the weight. So a lot of the ladies that were going into the facility that I was losing weight would be in disgust, because I'd lose 20 some pounds and they'd look and they're looking at their 5 or 3 or whatever it was. And what I've come to know, you always get your doctor's advice, but if you take vitamin D supplements, making sure that you have your USDA requirements of that vitamin D, you will lose weight as fast as the men. And it's pretty well known too that most Americans, I guess, or I would just speak for Americans, that we don't have enough vitamin D in our diet. In fact, they missed the USDA requirement number some years ago. They admitted by a factor of 10. Much too low. You actually need more than what they said. So I just thought I'd bring that up. So if you want to lose weight, vitamin D is good, and also For COVID-19, they say an increased intake of vitamin D helps, for whatever reason, to be a little bit more resilient to if you were ever exposed to the virus, in addition to zinc and a few other things I've already mentioned. So, but vitamin D is good for your immune system. So why is the difference? We're going to talk about that and what hormones do and various things. But vitamin D acts like a hormone and turns on a bunch of genes. If you don't have it, those genes don't get turned on, and some of those are related to the way you metabolize things. So just a tip from your Uncle Ed there. We'll keep that amongst ourselves as a secret. How's that? So mitochondria are found nearly in all eukaryotic cells. Mitochondria, the energy is contained in the form of chemical bonds, chemical bond energy itself, and as I mentioned, 32 kilocalories per mole. In other words, the amount of energy, heat energy is one way to evaluate as a calorie. It raises 1 degree Celsius, 1 ml of water, 1 degree Celsius is a calorie, and so that the energy that's in these are pretty good. It's a certain amount, individually low, but collectively a lot. And these are the really the pay to play for all the activities of our bodies, muscle contractions and forming various new compounds with enzymes and all sorts of things. Now, enzymes, again, if you recall, reduces the amount of energy it's required to either break or make a bond. But mitochondria also have their evolutionary origins as symbiotic bacteria. So if you were to look inside, to look at the genetic material, which I did not cover here as an image, but it does have circular, rings, a chromosome, just like bacteria do. And also its ribosomes. There's different weights between prokaryotes and eukaryotes. It's got the prokaryotic size, a smaller size of the ribosome, and we'll talk about all what that means later. But and it's also conferred via the mother. It confers the genetics for the mitochondria. But very interesting. I'll bring that up for whatever it's worth. But the mitochondria really help pay for everything. So, can cells change their composition to adapt to their environment? Absolutely. You may have been cold and you start to shiver. Well, we know that muscle contraction is not 100% efficient. About 40% of the energy is transformed in the form of heat, and so our bodies know that, and it's through evolution, and we start to shiver. And what you're doing is you're causing a muscle contraction and taking advantage of that 40% transfer of energy in the form of heat, and we can use that to try to stay warm. Your body will protect the core of your body. I mean, it's nice to have hands, fingers, toes, and all that, but when really cold weather and you're out there and you're not, you're getting exposed, your core gets the preferential treatment, and the extremities don't, unfortunately. But exposure to cold increased the mitochondria. Makes sense. You got to have the chemical bond energy. They're larger. The number of blood vessels double, so you got to move them around and get the ATP everywhere it needs to go, and reduces the size of fat cells. Why? For the very same reason I just told you, is the increased demand. So we got to convert a lot of that fat into hydrogens, cracking and peeling off the hydrogens to charge the battery to spin the ATP synthase. Does that start to make sense? I hope it does, because that's really cool. It's an appreciation. I'm totally in awe of ATP synthesis. Now, I've 3D printed it, so if by chance you would like to have a little model of an ATP synthase, I'll figure out a way. You can send me an email or something, and I'll see what I can do about sending one. Of course, my wife would immediately say, well, that's going to cost money with mail, so I haven't figured that out yet. But I do have a website. I'm thinking about putting it up there, and for a few bucks you can order a copy or something like that. I don't know, whatever it just covers the cost of mailing it and printing it. So we'll see. If you like to see something or have something like that, please let me know. just send me an email to acvinyl@waketech.edu, and I'll be glad to respond and see what we can do about that. And that's pretty much anything that we talk about. I can print any of the proteins we talk about. So uh, we have number of mitochondria and cold stress, and you look at the size of the mitochondria control versus the cold stress, and you can see in every case the numbers are large. Not only large, but significantly large. And so there is a correlation. And so in fact the environment does make changes in our bodies, and I'm sure there's not a student that's listening to this is not aware of that. And that's why a lot of athletes will exercise or perform their skills up in the mountains where the air concentration's lower and all those sorts of things. So when they actually perform at the lower altitudes, they'll actually have more reserve and more energy and all that sort of thing. So we've known that our bodies respond to the environment pretty well. So form follows function in organism cells and reflects their environment. Well, we know that. When cells must perform intensive heat production, they significantly increase the number and size of the mitochondria, because they're the ones that actually convey and utilize the energy by utilizing that hydrogen that drives the ATP synthetases. And in order to get that hydrogen, we had to cleave it out of fat. So there you go. They also increase blood supply, and that way we can distribute the energy throughout the body, and pretty cool. So there's that part. Lysosomes are essentially the cell's paper shredder. We shred paper to get rid of it and break it down and make it easier to put it in recycling and that sort of thing. But also, the lysosomes are the ultimate green machine. It breaks everything down to elemental components so it can be reused. And it's just very efficient. Our bodies are very efficient. So lysosomes are round, membrane enclosed, acid-filled organelles that function as the cell's garbage disposal. Yes, to some degree. It's more of recycling. Your book says disposal, but it does recycle. It does use the amino acids and the various building blocks. So they are filled with 50 different digestive enzymes, and dismantling the macromolecules, and also, of course, disease forming bacteria and viruses and rickettsia, whatever. You get the point. So endoplasmic reticulum, we talked about, build proteins and disarm toxins. So the smooth endoplasmic reticulum, the one without the ribosomes, really do this function. We mentioned the functioning of the liver. Well, you can imagine the liver cells would probably have a higher amount of smooth endoplasmic reticulum. In addition to the rough, they have to have both, but you would be in fact correct, because a lot of the toxic chemicals and cellular products, there's a higher demand in the liver cells, and so we see higher amounts of mitochondria and also these types of structures. And so it really, structure and function is just really interesting. Just to reiterate, we talked already about rough endoplasmic reticulum, but I just want to make sure that these studs and things that are on there, these are the ribosomes bound to the sort of the transport hole that we can get the messenger RNA through, and we make proteins, and then they'll go into the shipping, eventually go to the shipping department, the Golgi apparatus. But first we have to make them, and this is what the shop floor does. And so they tried to show you an electron microscope of what this represents, and it really doesn't show it very well, but trust me, those structures are there. And the transport system within the cell, just to reiterate, we have various different types of linked. There's the ribosomes and the messenger RNA going through the pores out of the nuclear membranes, and you can see it zipping, and the ribosome comes in two parts. It clamps together, and the messenger RNA is read and the protein's made, and it just starts to zip around. It travels on the messenger RNA about 100 miles an hour, so they say. I can't actually know that. And there you go. You see it goes through the pore. It's sort of like a little mobile 3D printer. Isn't that amazing? and the two subunits of the ribosome come apart, and then it's transported in the little microtubules of the sacs of phospholipid bilayers, and then the Kinesin takes that to wherever it needs to go. So the Golgi apparatus is the FedEx shipping department, or UPS, and then it melds with the membrane and releases whatever has been manufactured, captured in the bag of goodies that the Kinesin is carrying, to wherever it needs to go, and it just melds with the phospholipid bilayers of the membrane wherever it's at and dumps it, or it remains included into the membrane, depending on what the functions are. Isn't that just amazing? Our bodies are just amazing. So transport and moving on. So the smooth endoplasmic reticulum doesn't have the ribosomes, but I wanted you to think about, once it goes to the rough ER, then the protein may move and get maybe flame retardant or some other add-ons to the particular protein that's been built that is not supported in the rough ER, but is in the smooth. So in the hustle and bustle and loud welding and all that noise going on in the rough ER, we go to the smooth, and everyone's in their lab coats, and there's soft music playing, and they're adding all these additions on and getting it ready for shipping. And there you go. The liver cells are packed with huge amounts of smooth ER because the liver is primary site for detoxifying. So I had already mentioned that, but it's really, really important. How can long-term use of one drug increase your resistance to another different drug that you have never encountered? Well, what happens a lot of times is that there's similarities between one drug and another, and the liver gets really good at detoxifying. And if there happens to be some similarities in the chemical structure and function, maybe just altered function a little bit, then your liver is already set up to break down whatever drug that you're taking. So even if you've never encountered it, if it has a good amount of homology or similarity to the initial drug, you can see that the enzymes are all set to go, everything's manufactured and on its way. Antibiotics are that way. So a lot of times doctors will shift the antibiotics to a different class of type, and your body can't break it down, so it'll be bioavailable. In other words, it'll be more activity and blah blah blah. So anyhow, the production and modification of biological molecules within eukaryotic cells occurs in the organelles. We know that we use the endomembrane system, the endoplasmic region. Endomembrane just means you got this sort of foyer, this area, space that's contained by a membrane. In rough ER, it's shipped. The proteins that will be shipped anywhere else in the body are folded and packaged. It's rough because we have the ribosomes studding all over it. The smooth, we have lipids and carbohydrates that are synthesized, and they're added to or modified to those proteins that can't really occur in the rough ER. And so, as I say, this is where sort of you see the welding, and the t-shirts with the pack of cigarettes kind of rolled up in the arm, and it's kind of rough environment. Where the smooth is playing music, and folks in lab coats, and things are just different. It's a totally different environment. Not really, but think of it that way. And then the shipping department is that specialized Golgi apparatus. We've already mentioned it, but redundancy is the best way to keep hammering after you. And so this is where, of course, the FedEx delivery guy and the transport vehicles, all of these are phospholipid bilayers, by the way, just to make sure I haven't lost anyone. All the structures are phospholipid bilayers. That's how we form membranes. And the transport vesicle is a phospholipid bilayer. It just happens to be, this one's cut half. And that always throws my students off, because when they do these slices with the EM, it cuts it in half, and it's round, it's circular, and it's blibbing, and you saw in the video that kind of that sac of that phospholipid, but inside and studded in the membrane are the proteins that were made in the rough and perfected with a smooth, and then goes to the shipping department, and then it's delivered by the FedEx delivery guy. So hey, it's an amazing system. And the FedEx delivery guy, yes, it is very much like this, and they've come to know that this little uh, Kinesin is what it's referred to, and there's ATP it's required for each step, and it actually has a front and a rear Kinesin. And when it comes to an obstruction, like there's a cable or something in the way, it stops, and it literally rotates along that axis, and moves the sac away from the infrastructure, and so it can go and further on down. It's just amazing that the feet come down sort of like, if you ever seen those cement trucks with the second set of wheels or the third set of wheels that aren't really touching the road unless they have a full load of cement. I mean, that, can you imagine how much that cement weighs? Well, you can tell a cement truck that's got a full load because those separate wheels come down and hit the road. That's what happens with the Kinesins. If this needs to move over or back up, it has another Kinesin on the other side. So these feet go up, the other feet go down, and it does whatever it needs to do. If it's come to you that, how does it know? I can't answer that. There's a certain amount of communication that's going on. It has knowledge about where it needs to go and how to get there. And those are one of the things that maybe one of you someday will work out and add to the story. But it's fascinating how all these things work together. And it just doesn't happen. There's a reason why and how, as messengers and all sorts of things. And finding that out is kind of the fun. And so that's what's going on. And the pathways are laid out. Why these self-build and then self-break down. Again, I can't tell you, although that there is some sort of signal that occurs, but they auto build and auto break down, and it's quite amazing. I mean, it's a signal that does that, and then where it's built and all that, it's all planned. And that's where the Kinesin walks on, and then it's the military does that when it crosses rivers and things. It puts down tracks and all that. But the knowledge and information to do all that is it just blows my mind. In addition to, now I built a 3D printed a virus model that someone at Scripps Institute put together, and what I had to do was insert little teeny magnets at certain areas of it. It took me forever, but I did it. And you can put those in a bottle, shake it, and the virus will auto assemble. And it's driven because you have that cohesive force from the magnet. So I have magnets just lined up right, and it formed the virus. This is sort of what's happening here, that there's charge, and the size and shapes are universal, and they only fit one way. And whenever the signal occurs, however that occurs, it builds it. And then when it signals, it breaks it down and recycles everything. It's just amazing. So the big picture is that we have the flow from the nucleus. We get the messenger RNA, and then we process messenger RNA into the form of proteins. And then we move that, either it needs to go to the smooth ER, and they should draw an arrow through there because it's got to go through the smooth ER too. It's a ductwork. And then it comes into the back door of the shipping department, or the Golgi apparatus, right there, and through the back, and then it's shipped out through the front, just like most of the FedExes and things. I had a chance to work at a FedEx delivery when I was in school, and boy, did they work me 4 hours, and I was exhausted. But all you're doing is moving and binning. So everything has to go to a certain place, and it's sorted, and that's what's going on here. The analogy really does kind of work. And then it may dump it out outside of the cell or become part of the membrane. It depends on what's needed. And again, the information's all encoded on the nucleus DNA. So Golgi apparatus with another organelle within the endoplasmic system processes molecules synthesized within a cell, packages those who are destined for elsewhere in the body, all of which is designed or encoded by the information in the nucleus. That's just amazing. Plants, the sister company of the regular, is how I handled it in the analogy of the cell. You get a chance to look at that. It's just a business analogy. It helps you kind of relate all the structure and function that's going on. This is a spin-off company, and they use photocells. It was about the best I could do with the analogy. So it uses solar cells. The solar cells, being a plant, uses photosynthesis. We're going to talk about what photosynthesis is, but I'm not going to do it in such a way where you memorize a formula. I'm going to show you exactly what goes on at the nanoscale. It's amazing. And once you see it, then you see the interoperability of photosynthesis, and then how we retrieve that energy back and use it to form ATP. It's just amazing, and I hope you view it that way, because every time I go over this, and I can't tell you how many times I have, I still I'm just in awe of what goes on. And so just know that the plant cell walls have different structures because they're outside and they have to be weathered, and they still have to communicate, so they have to have specialized structures to do that. And I'm not going to have you name these sorts of things, but plasmodesmata, anything that mata or desmata refers to an opening, and it's an opening that goes to the plasma membrane. And see, that's the systematic way of learning that. I hadn't gone over that until 10 minutes ago, but the idea though is that the words do mean things, and this is Latin, and it does person, place, or thing at the end. And desma, of course, it's an opening, and in the opening where in the plasma membrane into the plasma. And so it's just nice that they use a systematic way of communicating science. I love it. The cell wall is an organelle found in plants. It's made primarily from the carbohydrate and cellulose, so we don't worry about so much the weather because we can put on a coat. But plants, they're kind of out there for the duration. If it's snowing or if it's hot sun, they have to kind of survive under these changing conditions, so it's prepared for the worst. It depends. I mean, you can have a plant that's a cactus and it has slightly different infrastructure, and we're going to talk about that some plants have to go through a dural cycle that's a little bit rougher than other plants, and so that's why we have some differences there. And then we have this plasmodesmata that connects the plant cells and enables them to communicate and transport between the cells. And it's pretty amazing. And then we get down, we're almost there. Vacuoles, just storage. If you could just equate vacuoles with storage, it would be really, really good. The types of storage kind of differs a little bit between cells, our cells, and plants. A lot of times, of course, plants are vascular and they need to store water in order to process the energy, and because they crack water, and we're going to talk about that, and that's where they derive their energy from the sun. And how it does it is going to blow your mind, absolutely blow your mind. You'll never see photosynthesis or think of it the same way ever again, hopefully. Waste management, predator deterrence. Believe it or not, they can store certain things in the plants that are naturally chemicals that ants hate, or some aspect of something in its environment, and stores it so that they don't get attacked or destroyed or whatever. It's pretty cool. Sexual reproduction, physical support. So vacuoles are storage. In animals, it's more fat, oils, and things like that, and not so much water. But in plants, of course, those are kind of roles. Now, I say it repels, but it also attracts. I don't want you to get wrong. A lot of times, certain pigments and things, we can go in and talk about plants. I love talking about plants. It's amazing. The colors of plants, flowers and things, but in the UV spectra, it changes totally. And a lot of insects look in the UV spectra, and so the image and how they go after things, it's just amazing. And if you ever get a chance to study that in higher courses, you won't be disappointed. So in plants, vacuoles, storage containers, can occupy most of the interior space. Vacuoles also appear in some eukaryotic species. In plants, vacuoles function as storage space, play roles in nutrition, waste management, deterrence, reproduction, physical support. So it's pretty much that. So getting down to the last two slides, chloroplasts are the plant's solar cell, the solar power plant. We're going to go over all this in photosynthesis, so don't worry about it, but I love just bringing it up where you have these thylakoids stacked up, and just like a nuclear power plant, you're dealing with a lot of radiation, that sort of thing, and you want special containment. Well, once I go through the explanation of how photosynthesis works, you can see why we need to contain it. And in fact, that's why they're there, the structures. Nothing's wasted in nature. There's a reason for everything. And just memorizing equations and showing photosynthesis, it's like a friend of mine said, it's like boiling chicken until the flavor is gone. So I don't want that to happen. I want you to have the appreciation of what's going on, and you can handle the truth, and you'll remember it, and it's just amazing. I want you to have that appreciation for that. So the chloroplast is the organelle of plants and algae that is a site of photosynthesis. By the way, bacteria, some types, can photosynthesize. The conversion of light energy into chemical energy with the oxygen as a byproduct. And you think about cracking water, what do you have? Hydrogen and an OH group. And so once you peel away the hydrogen, then the oxygen group becomes something you have to deal with, and it becomes oxygen. Chloroplasts may have originally been bacteria that were engulfed. It's the same thing. Their chromosome is like bacteria. It's like the prokaryotes, and also the ribosomes are the same type. They go by size. Eukaryotes have larger, and prokaryotes have smaller, and the chloroplasts have the smaller size. So it's obviously not from us. It's a guess that's come live permanently. So the cell structures. This slide is nice. It just shows you the difference between animals and plants. You know, that's always a question. The cell wall and looking at the chloroplasts are two of the differences. We don't have chloroplasts in animals, but we do in plants. Now the vacuoles, it says sometimes. And don't let that trick you. It's just fat is sometimes in the human or the animal cell, where the plant uses it for mostly water or storage of various chemicals and things. And in the cell wall, of course, we have harsh weather for the plants, where we don't deal with that. They're kind of the same sort of environment. So anyhow, point that out. You can look at each of those. We've talked about all of them, and that is chapter 4. And I hope through all of this that you got a pretty good appreciation of what's going on in our 100 trillion cells that we have, and the processes that are going on. So we'll pick up next time on the next chapter, but for now, thank you for your time, and we'll just keep